All right. We saw the overview of photosynthesis in two parts. The photo part is the light reactions, and the synthesis part is the Calvin-Benson cycle. And in the light reactions, that happens on the thylakoid membrane. So I'm going to put that on a structural perspective with the chloroplast. So here are the thylakoid membrane, and this is where the light reactions will happen. And the light comes in as an input, and it is mainly from sunlight. And there could also be artificial lighting that also helps and have the same effect. And there are proteins, complexes called photosystem. They are located on the thylakoid membrane. Okay. So that is what is going to help. And they are the photosystems. So these photosystems, 1 and 2, are located on the thylakoid. So when the light energy hits the antenna pigments, so the electrons get excited and they go through the electron transport chain. So these are the electron transport chain, ETC, and they end up with NADP plus and a proton, and that becomes NADPH. Okay, so I'm just reviewing what we discussed in the previous class. And while that's happening, we also noticed this concentrates protons inside, and the thylakoid become rich in proton when the electrons are going through. And when these protons come back through ATP synthase, so they are getting concentrated inside, and they come back through ATP synthase enzyme. It helps make ATP by combining ADP and PI, which is inorganic phosphate. Okay. So this is what is critical to understand. These are the ones going to go to the Calvin-Benson cycle. Okay. So, but where the electrons are coming from is where water comes in, where it gives 2 electrons to the electron transport chain, and it is split into 2 protons and half of O2, and this oxygen is what is released to the air. Okay, so I have completed the chart of inputs, light, water, and NADP plus and ADP-PI. All these are the inputs. And we saw the outputs are going to be the oxygen that comes out, NADPH that comes out, and ATP that comes out. Okay, so these are the outputs. Okay. Now, Let us see what happens in the Calvin-Benson cycle. Carbon dioxide comes in. Okay. It is now fixed into, so this is a cyclic pathway. This will become carbohydrate, CH2O. And to do this, we notice that this process is endergonic process. That means it needs more free energy. And it's an endothermic process. The potential energy is consumed, not released. And it is a reduction process. We need to understand all these. Okay. So because it is endergonic process, it needs ATP to happen. Because it's a reduction process, it needs NADPH to get reduced. So that's where you need to understand the logic, then you don't easily forget this, okay? So let us see what happens here. The NADPH, it goes in and it becomes NADP+. It gives that hydrogen to CO2 to make CH2O. Because this is an endergonic process, it needs energy to happen. The ATP goes into the Calvin cycle and becomes ADP and PF. So this is the Calvin, and I'm going to put this in parentheses. So many times just said Calvin cycle. Okay. Any question up to this point? Do you see the connection between what goes in and what comes up? So this is the sugar now transported via this structure called phloem. Okay. And this is all happening in the stroma, and the light reactions are happening on the thylakoid.
I have a question about the Calvin cycle.
Sure.
So the Calvin cycle can't happen until the light reactions happen?
Yep.
Okay.
Without this NADPH and ATP coming in, this is not going to go anywhere. They'll stop. So that is why in the olden days, they used to call this as a dark reaction as opposed to light reaction. That was a misnomer and wrong. And then they started using the word light dependent reactions and light independent reaction. That's also wrong. Many textbooks, you will see this as light independent reaction, but shut down the light, this is not going to happen. So it indirectly depends on the light, not directly, but without light, you don't have an NADPH, you don't have ATP, there's no Calvin cycle. Why you need NADPH? Because it's a reduction process. Why you need ATP? Because it's an endergonic process, meaning it consumes free energy and the potential energy. That is what makes this endothermic. Okay, good point. Any other question? Everybody good so far? So always come back to this big picture. You don't understand this big picture, you're using flashcard to remember inputs, outputs, blah, 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 blah. It's not going to stick. You have to find the logic and try to reason out your way. Then you try to reflect on it, you will remember better. Okay, so that's the idea. So let's go to the actual process of Calvin cycle. want to give credit to both the people. That's why wherever possible, I will use the word Calvin-Benson cycle. Okay. So what happens here is this carbon dioxide comes in, and it's combined with the 5-carbon molecule called ribulose bisphosphate. And this is a 5-carbon molecule that's combined with a 1-carbon molecule. They're supposed to make a 6-carbon molecule, but that is unstable. So they will end up with 3-carbon molecule, 3-phosphoglyceric acid, or 3-PGA. This is a 3-carbon molecule. Phosphoglyceric acid is what that PGA stands for. Okay, some terminology, it may be hard for you to remember, but think of it like PGA, a Professional Golfers Association, just happened PGA tours and all this national competition. All that, come up with something creative that sticks, and you remember that. Golf, that's a green golf course, and that's the first time carbon hydrogen combined to make this molecule. Okay, so this step is the most important step and the key regulatory step, and it's catalyzed by the enzyme Rubisco. Okay. So the enzyme Rubisco is an important enzyme, and we will talk about it next after I complete this cycle. So this fixing carbon dioxide from the atmosphere into a solid form is called as CO2 fixation or carbon fixation. So you're fixing an air gaseous molecule into a solid molecule. Then you need to continue into the next step. This is a acid. It has to be converted to sugar. So this is what creates into G3P, which stands for glyceraldehyde 3-phosphate. Now, an acid becoming an aldehyde, which is the sugar 3-phosphate. And when they combine, 2 of these molecules combine, it will make 1 glucose. And I will complete this and give you the number and the equation correctly, which is C6H12O6. That's a glucose. Okay. So this step of reducing this acid to an aldehyde, this phase is called reduction. So this is where the NADPH is used to reduce this, and it becomes a NADP plus. And this is an endergonic process. ATP is used, and that becomes a ADP and the PI. That's where the ATP and NADPH are used. So now, because it's a cycle, you have to regenerate the ribulose bisphosphate. Okay, so that phase is called regeneration of RUBP. That's how it regenerates that. For this, it uses an ATP, and it gives the phosphate to create this bisphosphate, means it has 2 phosphates, and it gets ADP. Okay. So you may wonder, one carbon goes, another carbon comes, or two, how does it all balance up? Okay, there are different ways people write this equation, but the ideal way to make one glucose, you have to put 6 carbon dioxide, and you have to have 6 5-carbon molecule that will make 12 3-phosphoglycerate, a 3-carbon molecule, and out of which 2 3-carbon molecules will combine to make one 6-carbon molecule, and the remaining 10 3-carbon molecules, that's about 30 carbon totals, that is going to become 6 5-carbon molecule. This is also 30 carbon total. Do you see where we balance all these? So you need to understand the overall how this is numbers are balanced, but you don't have to memorize, and I won't be asking you these numbers. Any question on the Calvin-Benson cycle? And we are going to look at this Rubisco in more depth. Then net inputs, what goes in, and net outputs, what comes out, is something important you need to remember. So the net inputs, carbon dioxide goes in, it becomes as carbohydrate, CH2O. NADPH goes in as a reducing agent, it becomes as an NADP plus and a proton. ATP goes in, it becomes ADP and a PI. Okay, so this is something important to understand. Question? And this is the process that makes the food for us. And everything else comes from this molecule. Storage. This is what becomes sugar, starch, everything, cell, and then structure, growth, cellulose, and everything. All these are important, and so that is how pull it up. Okay, so that's the source of all the carbons, and this also is the one that is going to become lipids, that we will see that later on, proteins, and nucleic acids. So you need to see the connection with all these things. Okay? And we will see how these differences happen later on. Okay, the big picture, the bottom line, and we'll go into this enzyme next. Okay, is everybody good so far? Any question? I'll try to upload the video as soon as possible today itself, so that anything that you missed, you came in late to the class, or you didn't understand something I said, you can always go back and rewind and fast forward, so on. Okay, so I'm going to continue this and complete the Calvin-Benson cycle. So we need to understand how important this enzyme Rubisco is and how it is regulated. Okay, so Rubisco stands for ribulose bisphosphate. That's the sugar, a 5-carbon sugar with 2 phosphates attached. And this enzyme Rubisco adds carbon to that. That's why it is called carboxylase and oxygenase. That's where the name Rubisco comes from. Okay. And so this is what the overall name of this enzyme. This is one of the largest enzyme complexes of 560,000 Daltons, 560 kilodaltons, and it's roughly 1,232 pounds per mole. Imagine that. Okay. So it's 560 kilodalton, or you can also say that it's 560 kilogram per mole. So there's another way to look at it. Okay, it's a humongous enzyme. It's like half a ton is the 1 mole of this enzyme. And 1 mole of glucose is only 180 grams, 180 grams. So it's very small, not even half a pound. So this enzyme is the most abundant enzyme in the world. So if we take the leaf protein, half of that will be Rubisco, and it's a slow and not so efficient enzyme. It's a kind of an evolutionary advantage of that. It's not so efficient enzyme. How is this regulated? So this is regulated by, and I will tell you why it is not efficient. CO2 and O2 concentration. Okay, these are substrates, and based on the concentration, they will go up or down. And regulated by pH, regulated by NADPH, and magnesium, which is a cofactor for this enzyme. Okay. But what is something to notice on this enzyme is a process called as photorespiration. What is photorespiration? So during hot and dry days, which is also called arid days, this is not hot and humid days, hot and dry days, the stomates, the pores on the leaf, close to conserve water. That's the main thing, to conserve water. Because of that, the CO2 and O2 exchange also stops. So when that stops, the CO2 concentration decreases. Why it decreases? Because of Calvin cycle happening, or Calvin-Benson cycle. And the O2 concentration increases. O2 concentration increases. Why the O2 concentration increase? Anybody? Because of what? This is, I'm talking about the O2 concentration increases inside the cell, mesophyll cells. Which process generates oxygen in photosynthesis? We saw that before. Light reaction is good. Excellent, Daniel. Light reactions, because of light reactions, O2 concentration increases. So too much of oxygen is also not good because it can cause oxidative damage. So then at that time, Rubisco comes to the rescue. It fixes oxygen and releases CO2 as a byproduct. That is what is called photorespiration. So it fixes CO2 into the RUBP. That's what it uses, RUBP, and releases CO2 as a byproduct, and an amino acid is formed. So these are all the part of the photorespiration process. It's like, and it is considered as a necessary evil. Why it's a necessary evil? If you don't do this, that too much of oxygen can hurt the cell and cell parts, and it's an evil because it wastes RUBP. You're breaking your own 5-carbon molecule and releasing CO2. So it is not a synthesis, it's a breakdown. That's why it's called an evil. Right? So how the plants have evolved to overcome this. Okay, so that's where we are going to C4 and the CAM plants. Okay. So they have an additional enzyme. So that enzyme is called phosphoenol pyruvate. That's called as PEP carboxylase. You can also call PEP carboxylase. That has no affinity for oxygen and fixes CO2 even at very low concentration. So that's the efficiency of this C4 plants. And there are variations between C4 and CAM plants, and I'm going to go through a comparison of all these. So the reason the C3 plants are called the C3 is because the first carbon molecule that's made is a 3-carbon molecule, whereas in a C4 plants, it is a 4-carbon molecule. And that's why it is called as a C4 plant. And CAM plants, it also makes this 4-carbon molecule, but it stands for crassulacean, that's a family name, acid metabolism. That's where the name comes from. Okay. So now let us understand, how these processes occur in C3, C4, and CAM plants. So they all have the Calvin-Benson cycle, which is also called as a C3 cycle that occurs in mesophyll cells and during daytime. Here, the C4 plants have a specialized cells called bundle sheath cells. They are next to the veins, and this also happens during daytime. In CAM plants, this happens in mesophyll cells. They don't have active bundle sheath cells, and in the daytime. Okay. So the C4 pathway is not present in the C3 plants, but in C4 plants, that happens in the mesophyll cells, the PEP carboxylase fixing the carbon dioxide into a 4-carbon molecule. And in the CAM plants, it occurs also in the mesophyll cells. They don't have the bundle sheath cells in the nighttime because these plants are desert plants where they live in a hot during the day and cold during the night environment. So they open up at that time. So the examples of the plants that are C3, C4, and CAM are rice and the wheat is an example. That's a staple crop. They need more water. And the corn and the sugar cane, they are very robust growing, and they can do more photosynthesis. And the CAM plants are cacti and pineapple. So they can live under hot and dry conditions. And so that's the difference between these 3 types of plants. Okay, we are done with the lecture. Any question up to this point?